Hi, my name is Rodhaini, and I'm from Watford Grammar.
My name is Natsumi Ishizu. I'm from Likya School in England.
My name is Genki Niwa. I'm from Momoya High School in Kyoto Prefecture.
The aim of our project was to produce a bioplastic from bacteria. So the first reason why we do this is because it's from a renewable source. For example, we can produce bioplastic from plants, and plants produce sugar and oil, and using that we can produce plastic. When plastic decays, it releases carbon dioxide and water into the atmosphere, and through the process of photosynthesis, it will be used up by the plant again, and the cycle starts again. So it's a renewable source, and we can guarantee that they won't run out. Whereas nowadays we still use fuel to produce plastic, and they're not a renewable source. They'll run out in the next 50 years or so. The second reason is it's biodegradable. As you can look at this image of a landfill site, a majority of the waste is plastic, and in the next 50 years, if you go to the same place and take an image of this, it will still look like this. However, with biodegradable plastic, the plastic would decompose in 2 months or so, depending on the humidity and the temperature. We also use bioplastics in the medical category, for example, for blood vessels and for during stitches as well, because of their properties as they're biodegradable. This is our main outline of our project and the steps we did. So the first step was introducing the gene into the bacteria. So what we did here was we produced a DNA with we modified a DNA which contained the gene to produce a polymer, and we inserted that into the bacteria. The second process was cultification. So basically we added nutrients that the bacteria required to grow, and we left it in an incubator overnight. As you can see, this image was taken just after we added the nutrients, and this image was just taken on the morning we returned, and this has gone a bit cloudy, so that shows that bacteria is present over there. And purification, so we purified our sample, and at the end we produced our plastic.
We need to introduce the DNA which make plastic into bacteria in this experiment, because usual bacteria cannot make plastic. So now I'll explain how to introduce the DNA in bacteria. First, we put bacteria and the DNA in a tube and put it in the ice. Next, we heated up this 42 degrees because this bacteria's hole opened and entered the DNA if bacteria was heated up. Finally, we cooled down this because this hole closed and put the DNA into the bacteria if bacteria was cooled down. So we can make bacteria which suit this experiment.
The next step was to produce the enzyme which produced the polymer. So first of all, the DNA contains the genetic code to produce the polymer. So we transferred the DNA to a similar molecule called RNA. The type of RNA is called mRNA, so messenger RNA, because it carries information from the nucleus to the cytoplasm, where the mRNA then interacts with the ribosomes, which reads the sequence of the mRNA, and soon the protein will be made. And the protein enzymes are a form of protein, and they then would have the instruction to produce the polymer, which is the plastic. Enzymes are a biological catalyst, so they speed up the reaction. And this is just an image of our enzyme. And when we inserted the DNA into the bacteria, the the process wouldn't be 100% successful. It'll only be around 30 to 40% successful. So, so there'll still be normal bacteria in our container. In order to kill the normal bacteria, we added antibiotics to prevent them from growing.
We grow the bacteria with special DNA, but we don't know really they accumulate plastic or not. To see if the plastic was produced, we use Nile blue stain. That is because the stain would specially bind the plastic. We used fluorescence microscope. We see in the two ways of light. One is the usual light and the other is fluorescence. Please look at the picture. It is a bacteria, and red area shows the plastic is produced. As this picture, we can find plastic in bacteria. Next, I'd like to introduce how to purificate plastic. First, we remove water and freeze dried bacteria. This is a dry bacteria. Second, add the chloroform to dissolve plastic from bacteria. Next, add the methanol to separate plastic and waste products. The picture shows before and after add polymer solution. In this picture, only plastic was purified. Lastly, we use centrifuge. It is a machine that spins container around very quickly so that the heaviest liquids and any solids are forced to the outer edge or bottom. We can condense polymer solution, and that picture shows practice that we made.
We use biological material for the research, but our laboratory is physical laboratory, so we use the technique or machine of physics to analyze them. For example, we looked at electron microscope. This is electron microscope. Electron microscope uses a beam of electron, eliminate a species, and produces an image which magnifies. The resolving power is greater than a light-powered optic microscope. The reason for this is because electrons have wavelength which is 100,000 times shorter than visible light. We use this and look at my hair. This picture is sample preparation. This is my hair. We can see very small parts. For example, this hair is 100 micrometers. Our teacher said my hair was good, so we can see very small things with this electron microscope.
Thank you for listening. And